This will just be the regular word. And the first one is sound check. But the first phrase is sound check. Check. So again, if you get words, that'll be a little extra credit. If you live streaming right now, words, you watching the video, those will just be your regular words. Anyway, here. Hughes is online. Fisher is online. Start from the top here. Abigail, Kincaid, Mullins, Cooper, Hunter. Oh yeah, Hunter's here. Here. Right there. Mel, I hear you. Gear. Julia, Talbert, Kennedy, Hannah, Larissa, Madeline. I already said Hughes is online. Galloway, Cook is here. Durgan is here. Is that WVSU shirt you're wearing? No, just checking. Okay, yeah, what I do, and this is good since I'm recording, this would be good for anybody. This is probably affecting a bunch of people. So what, for your lab, first thing I'll check is my emails to see if I got the email. And then if I don't do that, I go to my drive, because if you shared it with me, it'll show up when I search your name. And that doesn't mean you didn't accidentally unshare it, or I didn't accidentally unshare it, or the TA didn't accidentally unshare it. So yeah, thanks for letting me know. No. Just send me what you just said in an email. We'll see. I should have time between now and lab to take a look at it. Burton is online. Kinder, Jones, Canterbury. Actually, everybody here is accounted for in person. What do we have online now? Some new people. Hannah is online. Hannah online. Good. Cooper is online. I think that's everybody. Since it's 6 minutes, I won't do any important. I don't want to share any new information when no one's supposed to be here. So you should have 60 points of independent work by now. Technically, you can still get the 100 points, even if you haven't done anything yet. You still technically could, but we'll say this. That is still also technically great work. So if your plan is to turn in 100 points worth of work the last weekend this is due, you're out of luck probably because you're not alone in that plan. I'm not going to have time to grade 100 points worth on the last weekend. And I guarantee you're not the only one who's going to do that. A lot of people are going to send me a bunch of stuff on the last weekend, and I'm guaranteeing you I will grade at least 10 points of that because that's what's due. All right, on the last week of independent work, just like every week of independent work prior to that, it's been 10 points is what's due, right? So writing papers now. And I mean, yeah, papers now. Especially talk to people who have written papers. The grading is not straightforward. I lose points for that. You might think you have a full page. Turns out you only have 3/4 of a page, whatever the case may be. Yeah, so start doing that. Or you may have sent something that I just can't accept. You may have thought, but it's not acceptable. So yes, make sure you do your independent work. I cannot guarantee that if you turn it all in, at the end, I'll be able to grade it all. Any questions about independent work? All right. So remember, Just write about something about biology that you are interested in. That's the key. Writing about stuff that I'm teaching about, fine. Kind of waste of effort. It's like, here, I'm teaching you. Here's photosynthesis, and then someone reads, here's what I learned about photosynthesis. Fine. It's good to get other sources, but I want you to find something that you find interesting, hopefully. Make it a little bit less boring. It's about independent work. I think that's it. Yeah, so make sure you get it done, please. It's 100 points out of 1,000. That's a full letter grade. What are some other reminders? I don't want to do announcements. So here, I posted it yesterday. The exam 2 is now, you can take it anytime you want. They still haven't converted it to to do a Google form. So you click the link, it'll be a Google form, just like the study guides. And you only have 50 minutes to take it. So make sure once you open it, the clock starts. So once you click that link, be ready to do it in 50 minutes. You have this week to do it, next week to do it, and the early portion of the week after spring break, right? So you have 2 weeks to do it. If you don't include spring break. What else? Yeah, that's it. Oh yeah, and if you want more time to take the exam, like I said in the announcement, the written, make sure you email me.
I did both the videos last night. I know sent me an email.
Yeah.
So I sent you. I don't know if you sent it yet.
Not yet, but yeah, I saw the video. Yeah, thank you.
Oh.
What else? That's it for the exam. Oh yeah, like I said, if you need more time to take it, just meet me online. As long as I can tell you're taking a closed book exam, then I don't care how long it takes you to. What else for announcements or reminders? No, can't think of much. Got 2 minutes. Let's see. Miguel, not yet. And Kate is here. Andrea, not here. Lewis, not here. Cooper's online. Hunter is here. Caleb Crowder's here. Reinhardt is here. Isabel is here. Julie is not here. Wait, Talbert's here, right? No, Talbert's not here, correct? Yeah. Kennedy is not here yet. Hannah's online. Larissa's not here yet. Madeline is here. Hughes is online. Galloway is not here yet. Cook is here. Morgan's here. Tate is here. Burton is online. Kinder is not here. Jones is not here. Canterbury not here. Hobson is here. Fisher is online. All right, did I skip anybody who's actually here? 1 second. 2 people. All right. So Abigail and Kennedy and Larissa.
I'm here. Avery.
So Mullins is online. Okay. Larissa is here. All right. Other than the announcement, the reminders that I already said, that's old news information. Actually, one more old news thing. There's a couple of people who have really bad attendance, which means, I mean, at the very least, I'm down a letter grade at the end of the semester. But usually that ends up with them failing altogether, so not learning, so they're doing great in the exam. Point being, I'm turning to you guys. Maybe you guys don't show up. Talk to them as classmates and friends. But hey, you should. You get points for this. Good morning. So yeah, please talk to your classmates. They need to be here. Anyway, what else? Announcement. The other announcement is a lab announcement. It's already in writing. I posted an announcement yesterday that has lab information, and make sure you read your announcements. Started. I have to use my laptop because once again, the camera and the microphone are not working. Anyway, what we finished talking about was the cell cycle, and the last off with was this thing called G. So remember, the cell cycle is and the mitotic phase, right? And the interphase consists of G1, G2. So we learned all that. And then we learned about the mitotic phase, right? We learned all about mitosis and cytosis. And then once we were done with all that, then I was basically saying, all right, but there are some exceptions to the rule. This is what we call this. This is the G0 phase, right? So this is an exception to the rule. And we already left off. We already talked about this bullet point. Actually, before I get started, do the first official word for attendance, which will be, let's just say varies. First official word. Anyway, yeah, the timing does vary. I mentioned that last. I talked about how different cells, body, they have different schedules, so to speak, right? Some of them are constantly replacing each other. Some of them don't ever replace each other, and then everywhere in between. So here's the thing, though: the daughter cell has to get a parent cell. If you mess it up, like I said on Monday, and you're still going to get a perfect copy, right? So if you mess that copy up, then every copy after that is going to be messed up. So it's very daughter cell has to be an exact copy, right? We don't need these errors. We don't need these mutations. So that's why we have these control mechanisms. This is why this is where we're getting this G, right? This is where we're getting these exceptions to the rule. But now we're talking about the points which kind of control whether it's in G0 or if it's in G1 or G2, that stuff. And I'll go ahead and tell you now, even though I'm teaching this, I'm going to be very on the exam. So most of the exam, at least for this chapter, is going to consist of the different stages of mitosis and meiosis. I might have a couple questions about this, but if at all. Anyway, these control mechanisms, what do? They ensure the cell cycle pauses until favorable conditions, right? So instead of if the cells it needs to go to S phase, it's not that until things are proper, right? So it needs to have favorable conditions. You can look that up for independent work. That would be a real deep dive. That would be a tough one. When I say favorable conditions, what do I mean by that? There's so many different things you could look up to it. What exactly are the cells needing before they go from one transition to the next? Your text hints at it, but doesn't really get into it. Anyway, as you might imagine, these checkpoints are located at the end of G1, right? So that's that transition between G1 and S. And they're also between the G2 and the mitotic transition, right? So before you go into S phase and before you go into mitosis, basically, right? Those are the checkpoints. And we will discuss all three of them separately. So here we go. The first one we'll talk about here is this G1 checkpoint, right? So this is before. This determines if the conditions are favorable to proceed, right? And again, 100 biology major class, you don't need to know, which is why I'm just saying, I just leave it at that, right? It determines if conditions are favorable, but you can look that up if you want. What do we mean by favorable conditions? So a restriction point, I'll put an X next to that, just to can ask you what it's called or what else it's called. And this irreversibly commits to the cell division, right? So once it gets past, once it leaves G1 and goes into S phase, once it crosses into S phase, that's reverse, right? It can't go back. So once it commits to the cell division, it has to check for adequate reserves, cell size, and damage to the. And again, that's just scratching the surface. You need to know put a next to that. Well, I'll put a question mark, meaning I'm probably not going to ask you that. But if anything, that's what you need to know. Like, what are they checking? Reserves, cell size, and damage to the DNA, i.e. mutations. And then you go into the S phase and you copy it with the mutations, then you're going to mutations, right? So you want to be able to fix DNA before you move forward. So again, this is what you need to know, but if you want to, you can really dive deep into this. In terms of what? Exactly how big does this need to be larger, because it's slowly working on cutting itself in half. What about DNA? How does it recognize the damage to the damage to the DNA? Things that you can look for independent work. Anyway, the cell doesn't meet those requirements, then the cell does not go into S phase. And I will say this, I'm going to do this at the end, but right now I'll go ahead and do a spoiler for you. This is basically what cancer is, basically at a very basic level. There's proteins that control these checkpoints. That G1 checkpoint is that are attached to your DNA, right? So if you have a mutation in those proteins, then they don't have a different shape, or they don't have the right function, and they don't do their job. So in any of your protein, well, I shouldn't say any, some mutations in certain proteins process, then this process breaks down. And that's one of the ways that you can have cancer, right? If this checkpoint is going to automatically go into S phase, it's not going to check for mutations. And if it goes into S phase with those mutations are going to be copied, have even more mutations, right? So again, later we will stop and actually talk about cancer, but I'll go ahead. This is what this is leading up to. Any questions about the G1 checkpoint? All right, the G2 checkpoint. This is the thing that basically says it can or cannot go into the mitotic phase, right? So at this point, it's already done with G1. It's done with S phase, so it's done the DNA replication. And now it needs permission, if you will, to go into the mitotic phase. And what they're looking for here is for cell size and protein reserves, right? So the cell needs to be big enough, and there needs to be enough reserve of proteins, which makes sense. Because remember, we're going into we're leaving G2 and going into the mitotic phase. So that's the phase when finally the chromosomes are going to separate, and then there's cytokinesis is going to happen. We're going to pinch it in half and have two cells. So you're going to need a bunch of protein, and you're going to need to be big to do that. And then finally, G2 also ensures that the chromosomes are replicated and not damaged. So another independent work topic if you want to dive deep into this. That's it, right? That's all I'm saying. At the G2 checkpoint, we ensure that the chromosomes are replicated and not damaged. But first, how, right? I didn't say how that is doing that check, right? And what happens if it is not replicated or if it is damaged? Then what happens? All things you can look into for independent work. Though, if any independent work, I would not recommend writing about any of this stuff I've been talking about, right? If you haven't written any independent work, choose something. Why does my dog think when it gets wet, right? Go look that up. Send me a 2-sentence paper, get a few points, get some practice writing papers and seeing how I grade. Then the end checkpoint. This is near the end of metaphase. That's something you should know, right? Where, because obviously the M checkpoint is in the mitotic phase, M. But where in it's in metaphase, and that should be easy to remember, because M, right? So instead of thinking of it as the mitosis checkpoint, phase checkpoint. This is also known as the spindle checkpoint. This thing is that the sister chromatids are attached to the spindles. So of all of them, this one's the most straightforward. It makes right. So as you know from the discussion we had about mitosis, these spindles have to attach to the chromosomes because the whole thing about mitosis, at least the way we learn it, it's all about chromosomes moving around, right? So if you're going to move chromosomes around, well, that's going to be an important step. But again, I'm still scratching the surface. I guess there is some stuff. So how do we determine if the spindles, what happens if they're not attached to the spindles, right? Is it fixed? Whole process pause. You can look all that up for independent work. So that's it for the 3 checkpoint. So again, you have the one between G and S phase, or G1. You have the one between G2 and the mitotic phase, and then finally you have this one that's at the end of metaphase. So it's between this one and anaphase. Which makes sense, right? Because once you have anaphase, then it's too late, right? Then you've already begun the separation of the sister chromatids, and now they're full-fledged chromosomes, and now they're off and they're doing their own thing. So that there's some sister chromatids that were not attached, then you're out of luck, right? In the French, the language, but yeah, some cells are going to get too many chromosomes. They get enough, and we'll talk about that here in a few minutes too. So any questions about the M checkpoint or any of the checkpoints for that matter? All right. So again, I'll remind you too before we move forward. Most likely there won't be any questions about this on the exam. If so, there will be few of them. And more than likely, if I do have any, they'll probably be in the extra credits. So, speaking of which, for this is the exam that is opened as of yesterday, there are some extra credit questions. So a lot of the stuff that I said in lecture, where I said, you don't need to know that. You don't need to know it. But some of that, it could help you because it is extra credit. So study guides first and foremost, but if you feel really confident, you're like, man, I've got these study guides down, I'm good. Then start studying the textbook. Start reading some of that other stuff that I didn't, or I said you didn't need to know it, or I didn't dig into that much detail. So let's move on. Last portion of this chapter, and then today we'll probably jump into the next chapter. And in your old textbook, this used to be one chapter. I don't like it being separated. But anyway, let's talk about cancer cells. Earlier, when we're talking about these checkpoints, if something goes wrong with the proteins involved in that, then we have cancer. So let's talk about it. It's a disease of the cell cycle. You definitely need to know that. A disease of the cell cycle. That's what the problem is. Because as you just learned, as we just kind of scratched the surface, the cell cycle doesn't just go on autopilot, right? It's not just going and going. That would be useless, right? So you don't always need to replicate cells. So we have to control it. We have to stop and go signals, right? And if something's wrong with that, that's cancer. It is caused from a mutations in the proteins that control the cell cycle, which is what I was saying earlier when we were talking about the G checkpoints, right? Those checkpoints. There's literally to your chromosomes that kind of control, well, there's also other proteins. I don't want to get too specific. Let's just keep it really broad. There's proteins in your cells that have a lot of different jobs. Start-stop signals. And if there's a mutation in the gene, the protein, and that protein will be the wrong shape, therefore it won't be able to do its job. The cycle gets messed up, have cancer. Could have uncontrolled. And that's basically what cancer is. What stinks about this basic description, explain it like this. And it is until you start digging. As a matter of fact, a big I see when I see garbage on the internet is when someone says, oh, this thing cures cancer. Then I know right away I've got BS red flags flying because cancer is such a broad term. There's so many types of cancer that require different types of treatment. And when I see someone's cancer, like it's some kind of blanket statement, usually that's a big old red flag. Especially when they say, oh, big pharma is just they want you to be sick, money off of you. But you could buy my supplements and I only charge $100 a bottle, right? So yeah, the big pharma is trying to make money and they'll be quick to tell you about it. But then they'll also charge you money for their supplements. Anyway, soapbox, let's get back to it. Cancer cells. Cancer cells, the control system. Very simple statement when you look at it. Dig deeper, that's a very complicated statement because there's so many reasons why it not respond normally to the control system. The control system itself is very complicated. There's so many number of things that could be wrong. But because they respond normally to the control system, they just divide excessively. Hence that bullet point up there, right? The uncontrolled growth. Where it gets really bad is when this happens right here. It may invade. I've been looking on little videos about cancer cells. A lot of them, getting sick of them. But anyway, find the one of all of them, or at least the top 5. I'll share them with you. They're nice. They're short. They explain what. this. Let me put a question mark, meaning I don't know if I'm going to this exam. But we'll talk about oncogenes and proto-oncogenes. So proto-oncogenes that are responsible for the positive cell regulators. So these are just normal genes. So like I said, there's and the controlling the cell cycle, right? Those checkpoints. Well, the genes responsible for them, those are the proto-oncogenes. Right? Just regular, normal genes. The genes control the cell, right? Then we have oncogenes. So they're a lot like the proto-oncogenes, have been mutated. Because of this, they can cause the cells to become cancerous. They're usually not harmful. The mutation makes them, which is sad in a way, and in a way it is, because it causes die. But sadly enough, or oddly enough, having one cell die because it can't copy itself is having a cell that keeps going and going and going, right? Because once it copies itself, it's duplicated. So now you have two cells that won't stop going, and then they duplicate themselves. So you have 4 that won't stop, and then 8, and then 16, and 32, and 64, right? Yeah, so you should know that. If anything, again, with the question mark. If you were like a question where you need to know the difference between proto-oncogenes and oncogenes, again, both of them are genes that are involved with the cell cycle. Proto-oncogenes are completely normal, and the oncogenes are the ones that have mutated in such a way that it'll probably the whole thing happen. With that being said, that leads to the last bullet point. Sometimes these mutations can increase, right? Sometimes, like in this bullet point, this first one right here, not harmful because it's just messed up and stops. Sometimes we get this, right? Sometimes it's harmful. Sometimes it breaks down the whole thing and it goes on autopilot and it keeps going. So any questions about this slide? All right, then we have something called tumor suppressor genes. These genes code for the negative regulator proteins. Meaning, let me back up too. Sorry, I should have focused on this a little bit more. I should focus on these words. I didn't really explain this. Positive cell cycle regulators, meaning these are the things that are like, remember I said like there's an on-off switch or a stop-go signal, right? So if you're thinking about it that way, the cell cycle, again, if it has stop-go signals, then these are the genes that are coding for the go signals, right? Positive means go in this case. So these are the genes that say, all right, let's finish this checkpoint. Let's go into the S phase, on the mitotic phase, right? Those are the positive. Tumor suppressor genes code for the negative regulator proteins, right? So again, there's stop and go signals. The last ones we talked about were the go signals. These are the stop signals. These proteins can prevent uncontrolled division, right? However, if you have a mutated suppressor gene, that results in that have no checkpoints, right? That results in cells that have no checkpoints, right? Once you get rid of these tumor suppressor genes, once you get rid of the stop signal, then it just keeps on going, right? So basically what we're talking about here is there's 2 ways that things can get screwed up. You can mess up the go signal, you can mess up the stop signal. And again, when you think of oncogenes, you should think go signal. When you think of tumor suppressor genes, you should think stop. Anyway, any questions about that? The next slide is going to give a little bit more information on this topic. There we go. Your textbook uses a gene. You don't need to know this gene. It's just an example, but it's a gene called P53. You can read about it in the textbook. You can look even more into it if you want. But here we have a normal P53 gene, right? So we've got this damage to the DNA. So we have cell cycle abnormalities, something called hypoxia. You can look if you want. Actually, let me think about this. I don't want to talk about this and waste time. Now, I'm saying here, right, this P53, it's a tumor suppressor gene. So it recognizes there's a problem. So it either stops it and then fixes it so then it can get restarted, or it stops the process and says, well, we can't fix this. That's what that gene does. That particular gene, one of many genes that do a bunch of different things, that's what it does. It says, oh, there's a problem. What we're doing, it's either fix the problem or let's kill ourselves, right? And that's what happens. However, if there's a mutated P53 gene, right? So this is this is still a P53 gene, but it's mutated. It's not doing its job. So yeah, there's this problem, any of these problems, but this gene's not there to say, whoa, there's problems. Let's not go forward. So that gene's messed up, so the cell cycle continues, and then the cells can become cancerous. Again, this is just an example. When you're studying for the exam, for the next exam, exam 3, I wouldn't necessarily study this infographic. It's just a good example of how these tumor suppressor genes. Anyway, any questions about that slide? The next word for attendance will be cycle. Anyway, another independent work topic if you want to look into it. This is really trivial, but I'm just trying to give you guys a lot of independent work topics because some of you haven't written a single thing. This will be really quick and easy. What chromosome is that gene found on? So the gene is P53. What is it found on? Completely useless information. If you look it up, it's not going to help you in any way, but looking something up, and it'll give you a couple points of independent work. What gene is what chromosome is P53 located on? According to this source, it's located on this gene. So that's it. I'm going to pull up the next one, all right? Wish I had already. Chapter 6. Maybe it's already pulled up. While I'm doing this, does anybody have any questions about anything? All right. Let's see. Downloads. Search here for chapters. A long time ago. 7. I might have to do this spring 23. No, that's not going to work. All right, let me pull it off the website. This would be good, though. It's a recording, so for those of you who don't know where to find the PowerPoints, if there is anybody, you'll be able to see how to do this. No. I would have done this before we started, but I spent time finding that camera to work and the microphone. Here we go. Chapter 6, Chapter 7. Again, in my opinion, the same chapter. It used to be in your old textbook. Save as download this as PowerPoint. There we go. Sorry for the wait, guys. So now we're done basically talking about mitosis and the cell cycle. So we're done with that. We talk about the basis of inheritance. So basically we're going to talk about meiosis and some other stuff. Chapter 7, the cellular basis of inheritance. In my opinion, we're starting to get into more relevant stuff to you. Well, not everything, but a lot of the stuff prior to this chapter and the last chapter was getting you ready to learn stuff that's more relevant to you, right? Obviously, mitosis is relevant to you. The cell cycle is relevant to you, and it without understanding cancer. And unfortunately, almost all of us are going to be either us or someone we love. And now we're talking about inheritance. This is going to be really important. Either you're going to have kids one day or somebody you love to have kids one day, and this might be important for that reason. Anyway, let's talk about sexual reproduction again. And we already did this in the last, talked about the difference between reproduction and sexual reproduction. This is offsprings that have unique combinations of genes. So that's the opposite of asexual reproduction, right? Asexual reproduction is a copy of the parents. Sexual reproduction is a unique blend. It's very important, especially once later in the start talking about evolution. We could have it, but anyway, this has a lot to do with evolution. You'll see how when we get there. So yeah, you definitely need to know that first bullet point. You also definitely need to know this. Sexual reproduction depends on meiosis, because meiosis produces the sperm and the egg. And of course, obviously, requires fertilization, which is when the sperm and the egg come together. And I remember correctly, this is a picture from your old textbook. And it's a great picture because these kids, they do look like a mixture of parents, which again, produce a combination of genes from the parents. That's why, generally speaking, a person looks like a mixture of their mom and their dad. And of course, that's just look. That doesn't even show everything on the inside, right? There's genes and variations of genes you don't even see. But anyway, any questions about that first bullet point? All right, this is how this chapter is going to be broken down. Actually, excuse me. Different order, right? I don't like the way your textbook teaches it. I'm teaching it. I'm just doing it in a different order. The order they did it in, I think, is confusing. Plus, this is me if you read the textbook and you don't like the way they teach it, then the way I teach it, or vice versa. If you're listening to me talk and you're like, well, that doesn't make much sense, read the textbook. Maybe you like the way they teach it, the order in which they teach it. First thing we're talking about is chromosomes, because to learn any of this other stuff, you need to know what a homologous chromosome is. As a matter of fact, or two, not just in this chapter. This is one of those concepts that's going to stick with us. So the first thing you need to different individuals of the same number and type of chromosomes. You need to know that. That'll be important for a while. So every human doesn't have problems with their chromosomes, has the same number of chromosomes. So unless there's some sort of issue, everyone in this room has the same chromosome. You have this number, i.e. 23 pairs. Number will be the next 46. It's a number, not a word, but you need to know that. I think you need to know, not because I'm going to test you, I'm not going to ask you what this word means. Use it. Cell is a typical body cell. In other words, in your whole body, in the context of this conversation, you have two types. Your sperm and egg, whichever one you might have, and you have everything else, right? And everything else we call somatic cell. 46 chromosomes that I'm talking about, they can be seen in karyotype, which is this thing right here. It's basically a picture of chromosomes. The next slide I'm going to show you is a better picture of the chromosomes. But so far, are there any questions? Again, you need to know that we have chromosomes, and that's one of the things, right? Every species has the same number of chromosomes, unless there's something wrong. All right. That's what it looks like. I'm not going to ask you this, but I'm just going to talk about it very quickly. What's made or any of that. But to produce it, you've got to break open the human cell in metaphase of mitosis. Why metaphase? If we're taking pictures of chromosomes, why metaphase? Actually, forget that. Let's take it a step back. If we're taking pictures of chromosomes, why do it during mitosis? Why not do it during interphase? Think about the lab for those of you. Understanding you correctly, I think you are correct. But remember, when it's an interphase, it's just a big squiggly mess. You can get this chromosome from that. As you know, and as you need to know for the third exam, the chromosomes coil up in prophase, in all. They're coiled up, so we can actually see them. If you were to take a picture, you couldn't take a picture of a chromosome outside of mitosis because those chromosomes would be just a big squiggly mess. So yeah, that's why we do it in mitosis. If you want to look it up, specifically why metaphase, you can look that up for independent. Whatever. The point is, it happens in metaphase. The cells, so we have access to the chromosomes. They stain the chromosomes with dye. As a matter of fact, those pictures you guys see on in the lab, those are stained with dyes. And then they take a scope, and then they kind of cut and paste those pictures and arrange them in matching pairs by 3 chromosomes that humans have, and we all have them unless there's some kind of mistake. Chromosome 1. Well, I'll get there. So, any questions so far about what a karyotype is? At least one question on the study guide about a karyotype, at least one, where it's something like, which of these things can you diagnose with a karyotype? And right now, that doesn't make much sense, but when you learn other stuff, it'll make more sense. But basically, I'll give you the answer. The only thing you can diagnose with a karyotype is anything you can. Extra chromosome, we could see that. If there was a missing chromosome, we could see that. If there was a male or a female, we can see that, right? What we can't see is if there's some mutation in the P53 gene that we were just talking about. Can't see mutations. I can't see each individual base, right? All we can see is the whole chromosome. So that's all we can see is chromosomes. Here's another picture of a karyotype. This one's put in color. So again, there's your 23 pairs of chromosomes. The third one, sex chromosomes. We'll talk about that when we get there. That's what it looks like. Now, when you're looking at here, right, so you might say, oh yeah, that's the homologous chromosomes. That's chromosome one from the mom, one from the dad, right? Homologous chromosome. Zoom in, what you're actually looking at is, yes, these are homologous chromosomes, but these are sister chromosomes. Now, I will say this, you can even just forget about the fact that, I think you should just focus on the fact that these homologous chromosomes, these are two of the same chromosomes, right? You have two chromosome 1s. You have two chromosome 8s, so on and so forth. You have two of each. One of which you got from your mom, from her egg. One of which you got from your dad's sperm, right? When they came together, they gave you one of the chromosomes, and the other gave you the other. That's about the slide. All right. Here we go, homologous chromosomes. And you get one from each parent. This is very important. It's very simple, but you need to know this. This is very, very important. If you can't understand this, the stuff I'm going to teach you, especially after this chapter, won't make any sense. You need to know what homologous chromosomes are. So again, you have 23 pairs of chromosomes. You have 23 homologous chromosomes. And for each one of them, you got one from your. They are all generally the same length and centromere position. I didn't because it's a fact, but that's a fact that doesn't really help us later in the semester. So it's true, but for us, it doesn't matter. But yeah, just so you know, generally speaking, if you were to look at my chromosome 3, it would be the same size, right? The chromosome my mom is this size. The chromosome 3 I got from my dad is the same size. And here's where it gets even more interesting, in my opinion. And then if we were to compare them to your chromosome 3s, they should also be pretty much the size. And they also, and this is where it gets more important for quite a few chapters, these homologous chromosomes carry the same genes. So if there's a gene that dictates how hairy your back is, and it's located on chromosome number, and I'm just making something up. So on my chromosome 17, I have a gene that dictates whether you have a hairy back or not. Then also that's the same for every other human, right? Everybody else has the same gene. They might have a different version of the gene, but they have the same gene at the same location on the same chromosome, which is really helpful. Otherwise, it's insane, right? You couldn't really do genetics because you'd have to know everybody's new genotype and how theirs is different. No, it's all the same. We all carry the same mutations of the same chromosome. If we're talking about the same gene, these genes control the same characteristics. Really, this should be a sub-bullet point of that. But, somebody somewhere decided you're not sub-bullet point unless you have two sub-bullet points. So I followed that rule. But, yeah, we're carrying the same genes. These genes control the same characteristics. Like I just said with my stupid little hypothetical situation, the gene that controls back hair on chromosome number 17. Anyway, again, we have 46 chromosomes or 23 pairs. I've said this number a lot. I'm not big into numbers in this course, but those are definitely numbers you need to know. 46 chromosomes, 23 pairs. And then that itself is broken down. We have 22 pairs of matching chromosomes, which we call autosomes. And then we have the 2 sex chromosomes. Again, we have sex chromosomes, 2 sex chromosomes. We have everything else, which is called autosomes. And another thing you need to know, for mammals and sex chromosomes, males are XY, females are XX. You need to know, it'll be a test question. Male XY, females are XX. Those are for mammals. You can look that up for other stuff. What about reptiles? Squids? About plants? So many things you can look into, right? It doesn't have to be mammals. Independent work topic if you want. What dictates what makes a male fruit fly a male fruit fly? What makes a male Bradford pear tree? The plants will get a little bit more complicated. For an easy answer, stick with the animals. Even that's complicated too. It could be, but anyway, that's it. For chromosomes, right? It's a one pair of chromosomes, the same one. There's just different versions. One from your mom, one from your dad. You've got the same genes, the same characteristics. Very important concept. Let's talk about gametes and the life cycle of a sexual. The life cycle of a multicellular organism is a sequence of stages from adults of one to the adults of next. I'm not going to ask you that, and it is kind of counterintuitive because when I think of a life cycle of a human, I think to death. But in the context of this conversation, here we're just talking about the adults of one generation to the adults of the next. So it's a whole different way of looking at things, which is why I'm not going to test you on it. Well, one of the reasons. Anyway, having two sets of chromosomes, one from each parent, is a key factor in the life cycle. And that's what we're about to talk about. Here's something you need to know. I can't talk today. Something you need to know is that humans are something that we call diploid dominant, which means that all body cells can use chromosomes. So basically everything I just told you, there's a word for it. It's called diploid dominant. The fact that we have homologous chromosomes, right? The fact that we have two of each chromosomes, that means we are diploid dominant. That means most of our bodies, or all of our cells, if there's no problems with them, are diploid. Homologous chromosomes are diploid. However, there are haploid cells, and you need to know what that means. Haploid cells have only one pair of homologous chromosomes. And basically the ones that we know of, right, the only thing we talk about gametes, right? Sperm and egg are haploid. It's definitely something you need to know for the exam. Sperm and egg are haploid cells. The other cell in your body. And I always remember that because to me, diploid, that DI, I thought of that like two. Help me remember that there's, two chromosomes. And hap, and this isn't what it means, but for me, this helped me remember it. Hap sounds a little bit like half. Sperm cells and egg cells have half the chromosomes that the regular cells have. So that helped me. Anyway, I'm pretty sure you know from years and years of school that the sperm cells come together to make a baby. Words to it, because these might be new words for you. So let's have that conversation of things you already know, but using these new words. The haploid sperm fuses with a haploid egg in fertilization. And once that happens, then you're left with something called a zygote. So you definitely need to know that. There's going to be at least 2 questions about that on the exam. Meets with a haploid sperm to produce a diploid or diploid, however you want to say it, zygote. Know that. A lot of. I don't know if those are new for you or not. But for me, they were haploid, diploid, zygote. Those were all words I hadn't heard before taking this course. Well, a version of this course along. Yeah, about this side. All right. Here's a talking about. This is from a different text. This is from the previous textbook. Again, the life cycle is from the adults of one generation to the adults of the next, right? So here we have these adults. Most of their cells are diploid. There's regular old cells, right? So they go through meiosis to produce some gametes, some eggs and some sperm. Those things come together in a fertilization. Once that happens, we produce a diploid zygote. And then that zygote then goes through mitosis, becomes a full-blown human, and the process continues. What I don't want to worry about is this right here. I'm just not even going to talk about this. This 2n equals 46, 2n equals 46, n equals 23. I don't want to muddy the water. You can ignore anything you see in the PowerPoints, and in the textbook. We won't bother ourselves with that. All right, so here we go. This is how it happens, right? So we have this homologous chromosomes. This should look familiar. I showed you a picture of phase in the last chapter, and this is similar, except now we're focusing on homologous chromosomes and not just sister. So we have chromosomes. They duplicate in S phase. So yes, again, just like last time, you're left with sister chromatids. But now you're left with two sets of sister chromatids, right? That set and that set. All right, that's the interphase before meiosis. Specifically, as you should know by now, the S phase of interphase before meiosis. So then in the first meiosis, those homologous chromosomes split. Oh yeah, I forgot. I'm going to say this officially later, but unofficially, I'll go ahead and say it now. Meiosis is broken down into two. There's two sets of cellular division, right? So mitosis, which was the last chapter, they only separated the one time, right? Here they have. You definitely need to know. This is big picture stuff. We'll start adding details then later. But for now, the first thing you know is the thing we separate is the homologous chromosomes. First step. And then the second step, just like mitosis, we separate the sister chromatids. So know that before you know anything else. Before we start talking about the details of meiosis 1 versus meiosis 2, prophase, metaphase, anaphase, telophase of each one, before we even learn about that, that's the first thing you need to know. The rest of the details don't even matter if you don't know this. So first we separate the homologous chromosomes, then we separate the sister chromatids. Any questions about that side? All right, let's talk about the process of meiosis. Now that I've given you that little preview, let's talk about it. In meiosis, germ cells divide and produce haploid gametes. We already know that, right? Of course, I haven't really talked much about what a germ cell is. For this course, it doesn't matter. You can look it up for independent work if you want. What is a germ cell? Next to this conversation, all you need to know is a germ cell. That's the cell that you get before you make a sperm or an egg, right? That's the cells we use to make our gametes, our sperm and our egg cells. So meiosis resembles mitosis, but it has two major differences. So once again, you can study how you want, but my recommendation in this chapter, just like a lot of the other chapters, is study the big picture and then start adding in. All right, we're looking at big picture stuff. So the two major differences between meiosis and mitosis. The first one is the chromosome numbers are cut in half in meiosis, right? In mitosis, you get a perfect copy of your original cell. It's genetically identical. Not only does it have the same number of chromosomes, but they're exact copies of each other. In meiosis, this is not true because, the first thing is those chromosome numbers are cut in half. You might start with 46 chromosomes, right? But then you go through one round of division, and then you go through another round of division. So you actually end up with cells that have 23 chromosomes, not 23 pairs of chromosomes, but 23 chromosomes. Because again, the number of chromosomes are cut in half. So again, imagine, where are we at there? Let's just do an easy one. Let me think. 5, 10, let's say 10. Let's say there was an organism that had, or a species that had 10 chromosomes. Man, I hate this screen so much. Anyway, it goes through the first round of the S phase. So now temporarily it has 20 cystitids, if you think about it. But then those get split, those get cut in half because it goes through the first round of meiosis. So now you're back down to 10. Then you go down another round of meiosis, so then you get cut down to 5. So again, you start at your original number. I know this is really hard to read. You increase it, you double it, you cut it in half, so you're back to your original number, then you cut it in half again. So any questions about this big picture slide here? All right. So that number that I just used was just an example. But like that on the exam, I might make some other number up. I might say, okay, this species of whatever has, 20 chromosomes to start with. How many chromosomes does it have after the first round of S phase? And you might go 20, so double it, 40. And then I might, all right, how many chromosomes then does it have after the first round of meiosis? And then you might say, okay, 20. And then you might say, okay, how many chromosomes does it have after the second round of meiosis? So cut it in half again, and the answer would be 10. So very basic math. If you have any questions about that where there's math involved, it will be very basic like that where you do not even need, right? Just double it, cut it in half again. All right, the second major difference is something called crossing over. This is really important. Well, this is sort of important for the exam. That's important. There'll definitely be at least a couple of questions that involve crossing over. The crossing over is the exchange of genetic material between homologous chromosomes. You need to know that. You can see the pictures of it happening down there. Those chromosomes getting a little bit close. You got one from your dad, one from your mom. The little footsie down under the, and they swap left. Right? So this will be really important later. Like a mom chromosome with a chunk of dad on it, and you're left with a dad chromosome with a chunk of mom on it. And this, again, will be important very later, so a little later. But yes, crossing over happens. And I will point this out. This is just a random generic example of what happens. In all actuality, sometimes it happens more than once. So in this picture, we just see this one swapping of genetic material, right? But sometimes it happens more than that. Sometimes there's a smaller chunk than that, right? Sometimes it's a larger chunk. Right? Always happen just that way. Sometimes there's multiple, sometimes chunks. It's all very. You need to know what crossing over is. The other thing you definitely need to know about crossing over is this last bullet. Is that it happens during the first prophase of meiosis, which should be sort of intuitive. Not the prophase part, but if you really sat down and thought about it and thought about everything I've talked about so far, you probably should have been able to get meiosis. Because remember, what happens in the first round of meiosis? We separate homologous chromosomes. So obviously this isn't going to happen in meiosis because they've been separated, right? So hopefully you could have figured that out yourself. But yes, this happens in the first prophase of meiosis. The prophase part to me is not. It would make more sense if they were near metaphase, right? Because they all met in the middle. But anyway, any questions about the two major differences between meiosis and mitosis? Okay. So here we go. Here's what it looks like. This is chromosomes. They're all uncoiled, so it's a big squiggly mess. They're all in the nuclear envelope, which is still formed. You have these centrosomes, which haven't started moving yet, haven't started forming a mitotic spindle. That's interphase. Then we skip through G2, 1. And this is very similar. You can look if you want, but long story short, mitosis, except we're not separating sister chromatids yet, because this is meiosis 1. We're homologous chromosomes, right? That's the big difference. Everything else is the same. So I'm not even going to go through, like when I teach meiosis, I'm not going to teach because that part's the same. Chromosomes coil, spindles attached to the chromosomes. New chromosomes get separated. Chromosomes are completely separated. So prophase, metaphase, all the same. The only difference here is in meiosis 1, we're separating homologous chromosomes. We're not separating sister chromatids. Any questions about that side? And sister, word for attendance. We will finish with probably a good 30 or 45 seconds to spare. You guys go great with all that extra time in your day. And I'll see you in lab today or in class on.
I did my lab yesterday. Do I have to come today?
Unless I've left that said you don't have to come today. Yes, unless I've told you specifically, you said you do not have to come to lab today, come to lab today. Or some of you can't come to lab today, then you can't come to lab today. So you'll know you can't come if you're not supposed to come to lab.
Yeah, but like I haven't commented anything yet.
Yeah, you should. Then you come. Hopefully I'll have time between to do it.
Okay.
But this is why I was in last week. I said, get them done early. I need time to do it. Sorry. But I will try my best between now and 10 a.m.
We know for attendance on all.
Oh, shoot me an email if you don't.